and you have done some analytical result. Now your target will be how you will validate that. How it is possible? It is not too easy to possible. It is validated. Because during the process of validation, you must have to consider or collect some data, and that data must be real data. That has been mentioned. And after that, during the formation of the mathematical model, after that, your formulated mathematical model, your analytical results, your model validation considering all data, and also the result which you are getting after all this done, then you just have to project it. That means the intervention, obviously that should be revealed or focused in the society. Now, previous outcome, before going to your formulated mathematical model, whether there are any such type of work has been done or not, that should you have to recheck. So that you will get some knowledge during the reviewing process. And that should be reflected when you are doing all this research work. That I have mentioned here, that is the previous outcomes, that is combining interventions, targeted settings, impact of new technologies, and advocacy. All this must be reflected after completion of your research work. Now, just I give a little bit introduction which has already been introduced by Professor K. That is SIR. Little bit he has been mentioned. That is SIR. S means susceptible, I means infectious, R means recovered or immune, that is SIR. S, proportion of population, that is susceptible at time T. Proportion of A number, that is currently infected and infectious at time T. Proportion of N that is recovered during that time. SIR mode is used to predict the three proportions at different scenarios, and SIR model can be adapted to SI model, susceptible infected model, susceptible infected susceptible model, susceptible infected recovered susceptible model, and SLRI, susceptible latent infected recovered model, etc. This categorically can be reflected during the formulation of the mathematical model. In a nutshell, SI model, SIS model, SIRS model, and SLIR model. Now, basic reproduction ratio. Yesterday, so many of our students or researchers has been asked, or during the hearing of the lectures of different lectures, I have been informed that our students or researchers are very much associated with basic reproduction ratio. Though I have mentioned here, that is the number of people that one sick person will infect on average is called R0. Here are the maximum R0 values for a few viruses, that is, suppose here hepatitis, Ebola to HIV4, SARS4, mumps. So many people can be infected from a person, and yes, that is 80 people are being infected, or they have the capability to infect the other people, that is number of 18 or 16. Now, effect of vaccination. We try to give the vaccination during the process of infection or before the infection. That is infections for which the immunity acquired by natural infection can be assumed to the lifelong hepatitis A, varicella, mumps, rubella, and infected stages that is susceptible, latent, infectious, and recovered immune that is R. Just you have to follow here. These people are being vaccinated, S and R, and this is that has the direct effect, and the indirect effect, the people, those who are infected and these are the latently infected and actively infected, it has the indirect effect getting vaccination. Now I will try to little bit discuss about the how can we explain in 1984, initially first it was discovered that HIV, that means the human immunodeficiency virus. Human immunodeficiency virus, HIV. And in a nutshell, it is to be described as AIDS, acquired immunodeficiency syndrome. AIDS. And what is AIDS? AIDS to come into possession or something means I means immuno, D means deficiency. It is diseases or weakness in the body's ability to fight off infections and illness. And syndrome, it is specifically clarified by that is a group of signs and symptoms that occur together and characterize a particular abnormality. So these AIDS is basically clarified literally in that sense, and HIV virus, that all this actually AIDS. HIV enter when human immunovirus entered in your body. It does not mean the person being to be treated as the AIDS patient. When HIV entered in your body, I know everyone, you know about how it is to be entered in your body through illegal relation. Sometimes the syringe that inject your body, basically that is the blood transmission. During the process of blood transmission from one infected person to another uninfected person, if it is happened, then obviously human immunodeficiency virus, that is HIV, it is to be entered in your body. Now, it does not imply HIV when entered in your body that the people is to be treated as AIDS patient. No, it has a name, has a threshold. Beyond that threshold, when the HIV counting of HIV is beyond that threshold, then the people will be treated as AIDS patient. People will be treated as AIDS patient. That's why I have mentioned HIV is the virus that causes AIDS. Ultimately, if you take necessary action or event to kill the virus, if you treat the people, then such position will not be happened. That is AIDS position. Not everyone who is infected with HIV has AIDS. Everyone with AIDS is infected with HIV. AIDS is result of the progression of HIV infection. But I have told you, that is beyond that threshold value, counting of HIV, the people will be treated as AIDS patient. And thus, anyone infected with HIV, although healthy, can still transmit the virus to another person. What does it mean? It means that I am 19 years of diabetic patient. I have 2 stents in my heart. But still I am working, and some of you will not be understand that I am basically involved with health related activity. Because of I am maintaining all these issues. I have repeatedly telling your administrator that I need this type of food. I need this. I need this. Otherwise, I will not be run out my normal life. So, a AIDS patient, he has that capability. He is a healthy. It does not imply. If I touch you, I am a diabetic patient. It is not the transmissible disease. But the AIDS patient, only AIDS, he is very healthy, very healthy. But his blood is transmitted with an uninfected person. That means that HIV entered into the body. And to some extent, that people must be moved forward to such a position that is beyond the threshold value of HIV. He will be treated as an expression. If he do not take any necessary measurement or necessary action to explore that HIV into. Am I clear? Now, what is the how HIV entered? I am just telling you a story. We are the people. We have CD4 plus T cells. We have so many cells. T cells. T cells mean immune cells. T cells mean immune cells. CD4 T cells, CD4 T cells, CD8 T cells, CD8 in CD16, CD25, CD28. So many immune cells. But CD4 is the most essential T cell. CD4 T cell. How it is to be? It is generated in our body. We have a bone marrow. In our body, we have a bone marrow. Constantly from that place, this black cell is occurring. Constantly. This cell is occurring. And another place in our body, in immune system, that is called thymus. Occurring from bone marrow, this cell is to be matured to the thymus. Right? From thymus, this CD4 T cell is exerted, circulated in different parts, our total immune system. Right? We have considered this T cell. This is the T cell. Have you seen the antenna? Long before TV antenna, right? TV antenna. This is the like a sleep antenna. This is CD4 plus T cells, and this is the receptor. This is called receptor, like an antenna. Now just you have to follow me. This is the antenna, and this is the particle HIV. It is tried in our body from an infectious person. That is who is totally infected or may not be AIDS patient but HIV patient. Right? That when they are interacted with illegal relation or any kind of transmission, this HIV entered through chemokine receptor. This is is another term, chemokine receptor. Through chemokine receptor, suppose I am talking this, I have a connection with that mesh. So such type of receptor here, this is the chemokine receptor. From this chemokine receptor, HIV enters into the T cell, this T cell. When it enters to the T cell, what is its target? In our CD4 plus T cell, we have a nucleus. And that is the nucleus. This is the nucleus. He tried to capture the situation, position with that nucleus. He try and try and try. So many processes, considering so many processes. Lastly, he entered to this nucleus and just removed that position. He just stayed out in that way. Right? So, during this time, the RCP or in a general sense, just I am telling you, that HIV, that virus, and his, what can I say, that liquid actually sustained in nucleus, in the nucleus. And to some extent, it is remove the nucleus and take, that is unintegrated provider DNA. From this, it will sustain a long time, that is near about 145 days, it sustained here, and totally infect these CD4 plus T cells. Totally infect. When he integrated of that nucleus, HIV, to that total cell, particular that cell, is to be infected. Not only infected, again, from that infected CD4 plus T cell, so many virus is to be released. So many virus. So many virus. These all are the virus. These all are the virus. When this cell is totally infected, actively infected, in scientific language you can say it is actively infected, and it will take near about 155 days, 45 days to active in that particular position. That means actively infected. When that cell actively infected, again, that cell release so many virus, so many. Again, that virus in your body, what you do? Infect another uninfected cell. And it will be continued, it will be continued, it will be continued. After 6 months, you are not understanding. After 6 months, you will feel bad. You are feeling bad. You will go to the laboratory, when they will take your blood, they will get, it is to be tested, they will get the result, HIV entered in your body. Then they will give some medicine, vaccine, chemotherapy, all they will be taken. Systematically, if you do this, then your life will be sustained. Otherwise, to some extent, will be done. Okay, this is the story. Parelson, Professor S.F. Parelson, you are the head. I mean, outside of the country, don't disturb me. What's up? Huh? Oh, otherwise, you can understand. Now, this is basically the stone. This is the stone. Now, Professor Parelson first He was established in mathematical model in 1989. Fast mathematical model. It was a very simple mathematical model. And he has given during that time some prediction. But he was also a great medicinal doctor. Also was mathematics. He knows mathematics very well. Later on, after 89, a lot of researchers from different parts of the globe, they are doing research. If they are doing research, if they are doing research, whether the hate can be removed from our society, or the clinicians or experimentally, what they are established, whether the mathematician can give some extra prediction using that medicine or chemotherapy, that is now counting still enough. Am I clear? In that sense, we have done a lot of what I will show you. Already a book I have published on HIV. I'll give a simple example of that. That is, I have already all these things I have already mentioned you. And what I wish to tell you, that is HIV continues to reproduce. CD4 counts gradually declines from its normal value of 500 to 1,200. And here another part, if preventive medications not started, the HIV infected person is now at risk for manifesting secondary infections and achieving death. Most deaths occur with CD4 counts below 50. So all these points are very much required for clinical latency to know about it. Now, just I will let you know a simple mathematical model. Simple mathematical model. That is here X, Y, Z, which has been considered. X, Y, and Z. What is X? X is CD4 plus T7. X is CD4 plus T7. What is Y? Y is actively infected CD4 plus T7. And what is Z? Z is cytotoxic T lymphocytes. All these words I have used already. I have used CD4 plus T cells. I have told him that it is coming from bone marrow, or produced from bone marrow. It is matured in China, and then it is different organs in the immune system. It is circulated. I have told you again, infected CD4 plus T cell, that is virus return. It will take few months, and after that it is to be actively infected. That is Y. And Z, it is cytotoxic T lymphocyte. Just I am recapitulating it or mentioning you that when a foreign body, HIV is a foreign body particle. When it is entered in your body, our immune system will be activated. Why activated? To kill that particle, right? CTL, that type of cell who wish to try to kill that particle. That is CD4. There are so many biological features in that thing. I am not telling you about it, right? But the general conception is we are not biological person. That means we are not biologists. So we will not go to that part. Just this we have taken. That CD4 cytotoxic T lymphocytes, it will try to kill that particle since the harmful element entered in our body. with this seed population, we will try to make a mathematical model. Now, I have told you that CD4 plus T cell produced constantly from bone marrow. When it is to be produced constantly in a particular rate of BSC, we are considering here lambda. Lambda is the rate of production of CD4 T cell from bone marrow, which is matured in the thymus and circulated through the immune system in a different field. Okay. Another term we have used here, P into X into 1 minus X by TA. Already Professor K. Lotka-Volta model has mentioned. Logistic fashion, logistic form. What does it mean? We have introduced that. The general sense is the production of CD4 plus T cell, it is not to be produced unbounded. It is not to be produced unbounded. Suppose your cell is produced here every time. Okay? If it is if unbounded growth, then it is to be up. Cancer. Excessive growth of cell is to be treated as cancer. Excessive growth of unbounded growth. So we don't have to reach such a position that the CD4 plus T cell will not be grown or produced unbounded. That's why we have taken here P into X. X means CD4 plus T cells. 1 into this by this Tm, X by Tm. That means when Tm reaches to X, that term is to be abolished, right? So no, or the excessive growth will be shut down. Or the excessive growth will be shut down. Totally stop. Right? Therefore, we have taken this term P into X into 1 minus X by Tx. And this term is Px. Every living organism, they have a natural model. Man model. Today or tomorrow or after 20 years, I will not be here in that group. Right? So, in a macro level, I am a man. In a micro level, in a living organism, they have also a mortality. And that mortality rate, cell is micro level. We are in macro level. We are the population. Cell is also a population, but it is micro level. That is also a living organism. And this living organism obviously has a mortality. That mortality rate we have considered as D. So it will be deducted minus D. Right? And here also minus beta x1. What I have told you? That is X will release X CD4 plus T cell. When it is actively infected, then what we will do? We will do excessive production of IGAS. Excessive production of IGAS. Right? So that is the infected cell. When this infected person or infected cell will interact with an uninfected cell, they will be interacted with each other. They will, because in your body, infected cell always attached to the uninfected cell. Right? All cells are flowing through your immune system. So infected cell will interact or accumulate with each other, obviously in a particular rate. That rate which we have considered here, beta. So beta X, following the law of construction, what will be the term? Term will be minus beta XY, right? That beta XY obviously reflected in which population? Infected population. That has been done during. Again, A is the mortality rate of whom? A is the mortality rate of infected virus, infected cell. And this is KYJ. Minus KYJ means when the foreign body particle enter in your body, or CPL2, CPL try to kill it. Right? CPL try to kill it. We are considering that interaction to each other at a rate K. Z is interacted with the infected CD4 plus T cell. That is where the enemy. Now, why is now the enemy? Because he containing the infected virus. He tried to kill that cell. Okay? And considering the following law of mass action, the term will be minus KY. Right? And CTA, it must be generated from whom? Because he is interacting with infected cell. That infected cell generated whom? But he generated virus. That virus ultimately matched with whom? Infected cell. So ultimately cytotoxic nucleosides will be generated from the infected. And that's why the term here is what? That means when we are making or formulating the mathematical model of rate of change of cytotoxic lymphocytes, that is DZDT, obviously the term is different. And even this J has also a mortality rate. That has been revealed here. That has been revealed here. So now this is the nonlinear state of differential equation. Now it is a nonlinear state of differential equation. Now we have done it. We are solved it. Or our contribution in this work. 15 years ago, I have done this work. 15 years ago. Our contribution was only we have considered here this term. That is the holistic fashion. Because Carlson has not introduced the logistic fashion. We have obviously we have seen, we have studied so many literature, and we have thought that the CD4 plus pencil production will not be produced uncommon. That's why we have introduced it. And we have studied this and not going to. Now it has been published in 2012, 13 years ago. 13 years ago. It's a good work, and because it has been cited so many papers. What we did here, just I have given you a story. The antiviral drug treatment along with immune activator IL-2, a control-based mathematical approach of HIV infection. What we did here, to kill the virus or infected cell, a general medicine is called HART. H-A-A-R-T. HART. H-A-A-R-T. This is called HART. H-A-A-R-T. HART. HART means highly active, highly active anti retroviral therapy. Highly active antiretroviral therapy. Heart means highly active antiretroviral therapy. This herb actually a combination of that. Suppose when you go to the doctor, then doctor, if the doctor gives you the antibiotic, then obviously he will antacid or antacid. Am I right? When doctor will give you the antibiotic drug, then obviously he will give you a antacid. As because if you take that medicine, no other situation not to be offered for taking that drug. Heart, so it is a combination of heart is antiretroviral drug, a combination of, and this drug it is on its RTI, on its RTI, RTI, RTI means RTI, RTI, and another is PI. RTI means reverse transcript is inhibited. Reverse transcript is inhibited. RTI means reverse transcriptase inhibitor, and PI means protease inhibitor. These two drugs, all I try to give, even now, when a HIV patient goes to a doctor, the initial dose is herb, and that herb is basically a combination of two drugs on its reverse transcriptase inhibitor and PI is protease inhibitor. But it works. It works. RTI. It tries to kill the infected cell. And PI, it tries to kill the new virus. That is the new virus that is generated from the infected cell. The drug try to kill the infected cell. That's why. And PI, that means he tried to kill the new virus. In this world, what we did, we have considered a four compartmental mathematical model. And we have given two graphs. One is protease inhibited, and another is IL-2. IL-2 means IL-2 is interleukin 2 cytokines. It's a cytokine. We have given these two drugs, and we have introduced these two drugs in this position and also in this position. Cytotoxic T lymphocytes, the work of these two drugs is PI will kill the new virus. Our target is to kill the new virus. And another task was how we can activate our immune system, or we can try to more active our immune system. That's why we have given these two drugs. And these two drugs we have given in two categories. First, we have tried optimal, and second, we have tried impulsive way, independent impulsive way. We have seen that this is very much actively works, and give some prediction here how we give this drug that has been checked by this research one. Furthermore, a lot of works which we have did on HIV that you can check through my webpage. I am not going to another discussion anymore. These all are the works awareness program. Now just I will let you know the autoimmune disease. That was the infection disease and it is an autoimmune disease. Autoimmune disease. I have given you the lecture on the autoimmune disease in my first part of lecture. Autoimmune disease. What does it mean? It means that sometimes our body thought a foreign body part is inside in our body. Sometimes our body think that a foreign body inside, a foreign body particles inside in our body. Or it will be activated. Usually what ultimately the disease occur during this situation, that type of disease called psoriasis. Psoriasis. And basically the population dynamical model of psoriasis. First we have formulated population dynamical mathematical model on psoriasis. I'll show you that few minutes later. Population dynamical model of psoriasis. First we have formulated this one. And another model shared from Professor University of Edinburgh. He has formulated a mathematical model in 2008. And that one, it is very much density dependent work, but it was not a population dynamical model. No such mathematical model during that period has formulated. But later we have from 2009 we are working this disease. First we have formulated and last we have completed the work that I will show you, that the psoriasis or the disease psoriasis. So just I will let you know that is what is psoriasis. Psoriasis is a common chronic autoimmune inflammatory skin disease. Skin disease and characterized by T cells mediated hyperproliferation of epidermal keratinocytes. This type of skin disorder is characterized by macroscopic and microscopic skin alteration. So basically it is a skin disease that I will show you. Skin disease and this is the dead skin. You know that it is the keratinocyte layer. I will show you the all pictures. This is the outlook of psoriasis. This is main causes. Psoriasis seems to be an inherited disorder that it is passed down through families. Psoriasis probably occurs when the body's immune system mistakes healthy cells for dangerous substances that I have mentioned, and skin cells grow deep in skin and normally rise to the surface about once a month. And in person with psoriasis, this process is too fast and dead skin called build up on the skin surface. Now this is the psoriasis and clinical presentations well defined and sharply demarcated. This is covered by white Silverman scales, and this is distribution, the psoriasis. Globally, the affected pupils of psoriasis. This is the chronic plague of psoriasis. This type of disease, this is the chronic plague of psoriasis in arms. You can check gouty psoriasis. This type of psoriasis, flexural psoriasis, all these erythromatic psoriasis, these are the psoriasis. This is also the palmoplantar psoriasis, and this is scalp-psoriasis. Scalp psoriasis, or actually scalp psoriasis. This is scalp psoriasis. This is the psoriasis, male psoriasis, male psoriasis. All these are male psoriasis. So, and this is the psoriasis arthritis. Psoriasis arthritis. Now, considering all this knowledge, we wish to try formulate a mathematical model. In 2009, we have formulated the first basic mathematical model. This is the bone marrow. This is the Simon thymic processor. This is the thymus. And this thymus, basically, where the cell, T cell, is to be matured. And from this thymus, on T cell, this immune cell is to be released. That is called T cells. We have considered different T cells in HIV. We have considered CD4 plus T cell. Amalgamation of all these two T cells, actually CD4, CD8, CD20, CD64, all these T cells, naming as T cells, and another immune cells which we have considered here T cell, that is dendritic cell. Now, this is the healthy skin. It is non-psoriatic, and just you have to follow here. This is the scaly, and this is the psoriatic patient. The skin condition is this. Now, this is our first model in 2009. We had published, and I am not. This is the basic, simple mathematical model. That is, this is the T cell, this is the dendritic cell, and this is keratinocyte layer. Actually, keratinocytes is the causal effect of psoriasis. Keratinocytes is the causal effect of psoriasis. And this psoriasis, we have formulated a basic mathematical model. And because of, I have earlier mentioned you that T cell and B cell, T cell means immune cells, both are immune cells, and these immune cells are produced from the bone marrow, thymus. It is to be matured, and separately it is to be circulated in different parts of the immune system. So that we have considered A T cells and A dendritic cell, and K is not a immune cell, it is the keratinocyte cell population, right? We have the three layers, epidermal layer and dermal layer. Probably you know that upper portion is the keratinocyte cell population. Below that, epidermal layer. Below that, dermal. So all these are the skin. The keratinocyte, this is the dead cell. Below that, the keratinocyte cell layer population. When psoriasis occurs, it is to be reflected on your skin. Or I have shown you the picture. So, this is very much associated with the dendritic cell and keratinocyte cell population. This keratinocyte cell population ultimately the causal effect of psoriasis. That's why we have considered K. Now, T cell, it is continuously produced from where? Bone marrow, we have considered at a level A and produced at a level A. Again, dendritic cell, it is also continuously produced from bone marrow. So we have considered it is a B. This T cell and B cell, T cell and B cell, that is immune cell and dendritic cell, T cell and B cell interact with each other or accumulate with each other with respective rate, that is delta and beta. Considering the following law of mass action, the term will be delta ln and beta ln, and all I have told you, the all living organism has a natural mortality rate. So mu L and mu dash L are respectively the mortality rate of T cells and dendritic cells. Right? Now, these when actually T cell and dendritic cell at each other accumulated with each other, or to be focused, ultimately gathered in such a position where keratinocyte are exists. That tells keratinocyte so that this interaction must be upgraded or moving towards the keratinocyte cell population, and that has been introduced here. Delta ln plus beta ln, that is delta plus beta ln. We have considered that delta plus beta equal to beta. That is to be revealed here as beta ln, and it has the natural bond. So we have general study. It was the general study. We have in 2009 we have done visual, and later we have taken a simple graph that we have done. I am not going to this part. I am very much eager to show you incorporate the mathematical model. We all just 10 years, 15 years before what we did, and now just you have to see the mathematical. I'm not going to discuss the mathematical model. Just 1 month ago, this in mathematical bioscience, it has been published. It's so years. We have considered here the stem cell intervention. We are giving the stem cell. Now, globally, this drug, any kind of work, this drug is given to the patient. Any kind of disease. This we have used that not practically, even the clinicians and the experimenters doing their work on this, but not yet marketized in the society or globally, in the society as well as globally. That's why it is in a lab test, and the data, all this data we have captured from the different unauthorized, that is not the unauthorized, yes, that we have verified here, validated here, and 1.5 years to publish these works, we have explained on 1.5 years. So, what I am telling you that you are our student, researcher. You just have to think a little bit, but you try to make it in a to the sky. Sky is the limit. So when you are getting some conception, getting some knowledge, when you are studying anything in that I have mentioned how to formulate the mathematical model, you just have to incorporate. You first have to try to formulate a basic mathematical model, one by one, you try to incorporate all that issue in your mathematical model. So this is my basically, and we have got a excellent result in this work. That stem cell, if you give the stem cell impulsive, we have shown that psoriasis can be controlled totally, not to be eradicated. Absolutely, not to be eradicated, but it may be controlled. Right? Thank you all of you. Last slide, if you wish. This is all the first one, which I have. The first one, just roll out. The first one, which I have, it has been mentioned. Last 5 years, I have published on different articles that have been given. A few of that has been given. The main works which I have been published in psoriasis, in HIV, in leprosy, and the leishmaniasis. That is the vector borne disease. And this is so many works you can get it from this. If you go to my webpage, you can get it directly from there. And another part of my research, that is the production part. If I get time tomorrow, I will tell you about this. The production level, biological production. And if I get time, and this is the work, which is the disease, most of the work are disease related work. The fourth disease, HIV, leishmaniasis, psoriasis, and leprosy. These 4 diseases I have did and published so many work. If you go in my webpage and these work, just last 5 years, if you can go, put papers that we can stand. Right? Okay, thank you. Thank you all of you. All right. We will be happy so much for that very good presentation, and
Particularly, two diseases have been looked at. One exemplified, one COVID-19, and then the other Tantalus. Skin disease called what? Psoriasis. Okay. I think there may be some questions. Who you want to ask some questions, particularly regarding the model formulation. I know that Prof has dwelt so much on the formulation of that COVID-19 model. Sorry, HIV. I kept talking about COVID-19 HIV model. We dwelt so much on it, and I know that there may be one or two questions, clarify some things. This is not population-based model. This is in-host model where you have to deal with the cells within a host, not another population-based model. We are seeing how the interaction between infected cells and the healthy cells are happening. The CD4 plus T cell, and then the that one was there now. Yeah, so we saw how all that. Yes, it was. I don't. Yeah, was talking. So now we saw how each of the cells are being converted from like the infected cell CD4 plus T cell is being reproduced and then becomes infected as the foreign agent comes in, and then we also saw how that the immune system also tries to fight it, then capture them mathematically. That is model formulation. Obviously, it just showed us some of the results without showing the process of getting that results in simulation. But the formulation itself, I think it will be good that we interact to that, ask questions. Particularly, we have a mixed multitude here, people who are from the biological sciences, who are also mathematicians, and so it will make the discussion more robust. So please let's now ask questions. I have one. So I will give us number. And I don't want us to, I don't want questions to give rise to questions. So if you have a question, just signify it once and for all. If we have online participants who want to ask questions, please let us know. I have two hands already up, so plus my one, making 3, 4. Number 1, number 2, number 3, and that's all. Please come. Okay, number 4. Who else? Who's the person? Please your hand boldly. Number 5. Who else? Okay, so I will be number 6. Please can you help me with stuff? I don't want Chrome to stay for too long. On me. So I think we can still have him remind. Let's set my up for him. Oh, you give him the other one. There's another one. I will be using this one for audience. Okay, so let's get the first question. Okay.
Thank you, Prof, for this wonderful lecture. I have 2 questions. The one on HIV. Yeah. Talk about CD4 space, which is supposed to be the defensive mechanism for the body, and then the HIV cell comes to attack. And my question is, does one cell need to wait for it to be attacked, and then with the other one standing looking, and then they do not join to fight the HIV? That is one question. And then on psoriasis, the first model you showed, it has 3 equations. I didn't see interaction there. I don't know what you'll say about it. I didn't find interaction. Because usually we have one thing interacting with another to give rise to a disease or whatever. Thank you.
So far as my understanding, the participants asked me the question, that is the persons, those who are fighting with each other, one person is infected person, another is non-infected person, whether the HIV entered in his body. Am I right? Okay. So just I have mentioned in my data, HIV, suppose I have visited so many places in Europe, so many places. And in the park, I have seen the HIV or AIDS patient, not HIV, AIDS patient. AIDS patient seated in a chair in the park. Beside that chair, on another person who is purely general person among non-infected person beside him. And the infected person, he has written and a placard in his arms, that is, I am an AIDS patient. What does it mean? It means that if you touch with him, or even if the glass of water which he is drinking, in the same glass of water, if you drink, HIV will not be transmitted, subject to the condition. If the blood transmission is to be happen. That is, if the AIDS patient's blood in anyhow transmit to your body. And another thing is that mother breast milk. So far as my knowledge, I have seen that the mother breast milk can, in fact, not scientifically, it is completely proved that it is to be happen, but I have received so many reports that mother's breast milk can be, in fact, to his child, but no non-infected patients and infected patients, if they are touched with each other, it will not be infected, subject to the condition that till the blood is transmitted to the uninfected person. This is the answer of the question number 1. Number 2, that is in case of psoriasis, I have considered psoriasis here. Psoriasis is not an infected disease. Psoriasis, hello, you asked me the question and you are talking with another person. It is not good. What I am telling you, just have to follow me. Otherwise, why you have questioned him? And the another question which you have asked me, that is the dendritic. First, I have told you that the psoriasis is an autoimmune disease. It is not an infectious disease. It is not infectious disease. So no infected term is being involved in the mathematical model. No infected term is being involved. First, you have to know about the biology. Otherwise, you will not be able to formulate the mathematical one. That is why I repeatedly mention here, it is from my beginning of my lecture, that infectious disease, non-infectious disease, autoimmune disease, all I have delivered, everything I have mentioned. And just you have to see, in case of psoriasis, when I read the disease of psoriasis, the biological sequences of the disease, I have seen that it is an autoimmune disease. I have mentioned it repeatedly. And our immune system thought that a foreign body particle has entered in my body. But no foreign body particle has not been entered in your body. During that time, Our immune system is to be activated and they interact with each other. That interaction is T cell and dendritic cell, and so many cells are there. That last part which I had mentioned, there's so many cells are cytokines are very much associated with this. Later we have started this work in 2009. Now it is 2026. At least 40 research articles have been published. This is the last research article which I have been published in mathematical bioscience. So that was my first mathematical model. My first mathematical model, dendritic cell, T cell, and keratinocyte cell population. It was the simple mathematical model, right? No independent term was involved in that mathematical model. Okay, thank you.
All right, thank you.
Thank you, sir. So we'll now go on to the next person. Thank you, Prof, for that beautiful lecture. And my question is on the HIV model. So I want to ask, in a situation where, let's assume, I know now, as at now, there is no HIV vaccine. But
Yeah,
as at now, there is no HIV vaccine.
I'm not. Yeah, I think
There is no HIV vaccine as at now.
vaccine as of now. Okay.
So, but just for some other viral pathogen. Now, how can that equation for HIV be modified if you want to introduce vaccine? In case we have to model another viral disease, which is not HIV this time around, that has vaccine already. So can we modify this HIV model to also capture vaccination?
The thing is that you can do anything. But the question is whether it is realistic or not. You can make the logistic fashion term in any mathematical model. But the question is whether the logistic fashion term is validated in that mathematical model. It is the question. Right? So if you wish, you will do that. Therefore, I have got a lot of papers. This term introduced here in this term. No. First, you have to know about the biology or the immunology in that particular system. Yes, you are right. No, vaccination is yet not been. Already, Russia has mentioned that they have already prepared a vaccine, but not yet marketed. But still, the same cell, which I have mentioned my last part, the same cell, it has been in the lab, it has been cured, that the people has been cured, but has not marketed. It is under the process of research. So the I wish to incorporate the vaccinated term HIV. No, you will not do that. The experimentalists, what they have used, you just have to use that thing in your mathematical model. Otherwise, no one will consider your own. No one will consider your own. Right? So
Yeah.
first you have to, before formulating the mathematical model, you must have to do the review of the work. Review of the work. What type of work has been done till now, then you incorporate that, you try to formulate your mathematical model. Okay, thank you.
Thank you so much. So that's very clear. You can decide to do anything if you want to do, but you must also be careful that it agrees with the reality, with the biology or immunology of what you are dealing with. If not, nobody will consider your work. So it's just being mathematical exercise. All right, thank you.
Thank you very much, sir, for your efforts in delivering the lecture. My question is on that HIV model. So you mentioned RTI that you introduced into the term that it will kill the infected cell. Then you mentioned PI also that will kill the new virus
cell being generated. So now, you talked about the CD4 being produced. And we know that the patient is already AIDS, has HIV and AIDS. Will the CD4 production be optimal? Will it still be producing optimally, having been infected? If it is not being produced optimally for the distribution to the bone marrow, why did you, or is it possible to now introduce a term that will enable, like the logistic term we introduced, that we should introduce a term to the CD4? That's what I want to ask. Is there a term that can be introduced to the CD4 just the same way we introduced RTI to the one that will kill the cell and the PI that will kill the new virus?
Okay, I was trying to follow him to understand his question, but what I can pick from the question now is that the CD4 cell, you talked about the CD4 cell. So he's saying that once the CD4 is already infected for an infected HIV individual, so will the CD4 cell production be optimal? Now, if it's not going to be optimal, is there a way to model that condition in the individual to track the fact that it may not be optimal, there may not be optimal production of CD4 cell? That is its own way of viewing it.
Just to have to recapitulate the essence of the CD4 plus T cell production. Essence of the CD4 plus T cell production, that means CD4 plus T cell produced from bone marrow. It is mature in the thymus. Then it is to be circulated through your immune system. Right? This is number one. Now, when HIV infects the individual, CD4 plus T cell count obviously will be decreasing. Obviously will be decreasing as because of the number of production of CD4 plus T cell. One portion of that is to be infected. Chronologically, it is more and more infected. The production rate is the same. So obviously it is to be decreasing. The cell is to be infected as because the virus is produced in your body. Because infected cells again produce the virus, again produce the environment. In my figures I have shown you, CD4 plus T cell, when it is to be infected, that uninfected CD4 plus T cell is to be produced. But in more and more HIV, when it is to be formed in your body from infected cells, that CD4 plus uninfected CD4 plus T cell will be reduced. Now, in that case, we have tried IL-2, the cytokines, that is an injection which we have introduced in the body, impulsive way to infusion, and the CD4 plus T cell count can be enhanced. Again, another drug, that new virus, those infected cells, those who are getting the new virus, that drug PI, protein inhibitor, that we have used in our mathematical model, and that drug is given to the newly infected virus, newly produced virus. So these two mathematical terms we have used considering the biological eventuality. That we have incorporated and we have given it in impulsive way.
Thank you so much, sir. So that in essence, he has just helped us to see that the HIV virus does not affect the production of CD4 cell that is produced from the bone marrow and it comes into the system. So it's in the blood system that the HIV infection attacks the CD4. So it's not in the production. So it does not affect the production. Thank you. So, okay, that's number 3, right? So number 4. Where's the fourth person? Can nobody from this end again?
Finger.
Thank you very much for the amazing lecture. I want to ask if you can have access to your slides.
Okay. So many people have asked the question, the slide, can they get it? Huh? Slide, your presentation slide, can they get it?
Sure.
Sure.
I'll give you everything, don't worry.
Please can you put your hands up?
Everything, everything. Everything will be ready.
So, thank you so much, Prof, for the lecture. So, my question is on the HIV model, the XYZ. So, you explained that we have a kind of constant recruitment rate, the lambda, then plus the logistic term in to capture the fact that the cells cannot be produced unboundedly. So in order to impose a kind of bound in the production of the CD4 cells, so you introduce that logistic function. So then just want to be clear that's why the addition of the constant term and the logistic term, knowing that if the I'm just thinking that if the logistic term comes, since it's also talking about maybe the recruitment of the new cells into the X compartments, is giving us two things at the same time, the production and the bound that we are interested in. So I just want to know the rationale behind putting the lambda there, having the logistic term. Thank you.
So Prof, before you answer that question, because that was exactly my own question too, that looking at the recruitment, we have lambda and we also have the logistic, as I said. But I look at the other model, which has to talk about, I'm trying to learn that disease now. It's a new one, psoriasis. For the psoriasis one, for the paper you just published, I realized that you just you stuck to the logistic growth alone without necessarily putting the constant recruitment rates. So I was also having that same challenge that while in the first one, including both the constant recruitment rate and then the logistic, why can we not stick to the logistic, since we know the logistic captures the unboundedness in terms of the production of the CDC. So, I don't know if you got the questions, sir.
That so far as my understanding that in that particular model, it was lambda plus Rx into 1 minus H by Tm. Your question is, the production is continuous in a rate, in a particular rate. The rate is being considered in that particular equation, rate. No variable is being involved in that.
Yeah.
Right? But in the second term, R into X into 1 minus X by T.
It is per capita growth rate now.
Am I clear?
Yeah.
Huh? Am I clear?
Yes. Yes.
So that was the rate. Your question is, why you have given an additional term? That is Rx into 1 minus X by K or TL, something like that I have written. So lambda is the rate of production of that rate. Whom rate? The rate of X. And rate is X is, it's purely mathematical. And X is very much associated with the production of that.
Sorry, that they were passing some information. I'm sorry. Please, after now, I will listen to you. Prof.
So Rx into 1 minus X by K, that's why I have given this now. That production is not to be unboundedly করবো. That is revealed that production, whom production? X production. X term, it is very much associated with X. Just you have to follow R into X. X means CD4 plus T3.
Yes.
That X is not to be put to unbounded.
Yes, sir.
And lambda is the rate. No X is associated with that. Okay.
Okay, thank you so much. I think somebody wants to say something. Please, you can move forward, sir.
Thank you very much, sir. I want to get more understanding. The fellow who asked the question about imputing CD4 count into the model. You talked that you said it's a biological process, but I also understand that we could evaluate how healthy an infected person is by knowing its CD4 count. So if your CD4 count is low,
If you, just I am telling you, if you have any biological question, you just have to tell me. It was the initial or suppose it was this was before 15 years. Before 15 years, so many things now have been involved. Latest mathematical model. If you have any biological question, you can ask me.
the
It
question
is a very simple mathematical. The basic concept was the model was established by the parents and I just incorporated simple term logistic fashion. Nothing needed.
the question I want to the question is this: is it possible to incorporate the value of the CD4 count to ascertain, imputing how healthy the patient
It
is?
is the general thing that no living organism cannot grow unboundedly. No living organism cannot grow. Suppose no living organism cannot be grown unboundedly. As for example, what is the example? Cancer. Excessive growth of cell. Its growth is unbounded. It to the reach the people don't know where it is to be stopped. That's because of the fertility. CD4 plus T-cell is the same. It is not grown unbounded. It is a general thing. So no living organism, your population, counting of population in Nigeria, it is not to be grown unboundedly. That's why it has been incorporated.
Prof, I think he's asking a different question. He's a biological science person. So the question he's asking now is that he's saying that CD4 determines how healthy somebody is. According to what he's saying now, that CD4 counts determines how healthy a person is. So he's now saying that our model, it's not a mathematician. So he's listening to us as a mathematician. He wants to know maybe our mathematics is agreeing with his own biology, knowledge of his biology. So he's saying that he knows that CD4 counts is a determinant of how healthy a person is. So he's now saying that
We cannot count it. We the mathematician cannot count it.
okay, so I think during the launch, some of these things will also be sorted. I can see that we may need to launch into another rate after now, so that in the period of the launch, so you'll be launching the other rate together. Is that okay? So please let's end this one now.
Yes, I'm talking a long time.
Yes. I need so many of the other discussions can be sorted while the